Hello, Kevin.
Hi, good to
Hi.
see you, Natalie.
It's wonderful to see you. We have been friends for a long time, and I'm so glad to get a chance to chat with you. Would you mind just introducing yourself and where you're at, and we'll go from there?
Hi, my name is Kevin Solomon. I'm an assistant professor of chemical and biomolecular engineering at the University of Delaware.
University of Delaware. What a great, great place. And wow, that's quite a department. So seems like it does a lot of things. What does your lab do?
So my lab is very much interested in domesticating the biocatalysis of environmental microorganisms. So to kind of put that in context, we're interested in addressing challenges in sustainability. So trying to think about how we can turn renewable plant material into biofuels, as well as how do we address this growing problem of plastic waste. And what I've realized over my career is that while synthetic biology offers a lot of potential to engineer microorganisms, some things are a little bit more challenging. So to use an analogy, um, while we can rebuild our cars, you wouldn't turn you wouldn't turn a Honda Civic into a race car. I mean, at that point, it's a brand new car, and so we
Right.
so we try. So what we try to do is we try to find race car like things in the environment, and then we use synthetic biology to tweak it to make it more amenable to societal applications.
That's so great. So it's like finding a nearest neighbor for what you're trying to get to. And I will just add that my son would love it if we could turn a Honda into an F1 car. Just saying.
People do try, and they spend a lot of time and a lot of money doing it. But you could buy a Formula One car that, I mean, granted, it's a little bit more expensive, but it's a lot more efficient.
Yes, yes, certainly the shorter distance to your endpoint would be to start with an F1 car or something close to an F1 car to get there. So that's very cool. So tell me who's in your lab and maybe is there something that you're super excited about that they're working on right now?
Yeah, so one of my senior grad, sorry, a new student in my lab. This is a new collaboration with another professor at the University of Delaware, where we're studying how mealworms, how they're able to degrade plastics. And so you might ask a question: why do mealworms even degrade plastics? Right. So mealworms are insects, and essentially they eat leaves. And if you look at leaves very carefully, they're very waxy, right? And so those waxes that keep the leaf waterproof and prevents the plant from losing too much water, those waxes very much look like the plastics you might find in your styrofoam containers or your water bottles. And those are the same kind of plastics that we don't really have a good mechanism of recycling them. And so this new rotation student, in a very short period of time, his name is Ross Clower, he has isolated a very vast repertoire of, so looking at different microbial communities, different microorganisms that are able to degrade different plastics. And now we're even beginning to pull down specific enzymes that we hope will lead to efficient strategies to break these materials down.
That is amazing. I will never look at a leaf again without thinking that it's got this waxy waterproof coating,
Yeah,
like a Rain-X for it or something. That's
that
very,
is
very
exactly what it is. It is Rain-X for
yeah,
plants.
for plants. That's so cool. So just to get a little bit nerdy about it, and then is it a microbe in the belly of the mealworm, or is it the mealworm itself that has those enzymes?
So it's a collection or microbial community that live in the belly of the mealworm that do most of the work. And so when we started off this project, we had these mealworms, and we could see them eating the plastic, like the plastics slowly grow over time. But then one of the key questions that we had was, well, is it kind of like corn? You eat corn, but if you kind of look, if when you go to the bathroom, you can kind of see little pieces of corn.
You haven't
But in
really
fact, that is not what happens. The plastics that come back out are very different. They're chemically modified. And since then, we have extracted the gut contents. So we've essentially collected all the microbes that grow in the gut. And we can actually grow them in isolation, and they continue to degrade these materials. And so now we are using the tools of systems biology. So we're essentially looking at the genomes and transcriptomes. So looking at the genetic potential of these organisms to try to understand what exactly is causing these things to degrade.
It's amazing. It sounds so truly interdisciplinary, which you hear a lot as relevant and important for solving these complex systems. But between the systems biology and the microbial ecology, and then the enzymology and everything else that, what were you trained in that led you to be able to do all this?
So during my PhD, I was working with E. coli like most synthetic biologists do, and I was really excited about the potential to kind of reprogram organisms to do things. And there's lots of things that we can do, but what I realized towards the end of my PhD is that some phenotypes are very complex. So synthetic biology is very good at putting in 1 or 2 genes. You can make it a little bit more sophisticated, but relatively speaking, compared to natural systems, those systems are kind of simple. A lot of these more challenging things require hundreds of enzymes. They have to be coordinated in a specific way, and trying to recreate that is non-trivial. And so I realized that a lot of what we, as they say, like in Jurassic Park, life finds a way. And so a lot of the things that we wanted to do existed somewhere. The challenge was that they existed in an exotic environment, so in a deep sea vent or in the stomach of some exotic animal that we hadn't looked at before. And the challenge was then, how do we find it? And then how do we make that technology accessible? Because we're not going to grow them in the guts of worms, or we're not going to grow them at high pressures in like you might find in a volcano. So how do we then take these systems that work very well, but then do it in a context that allows us to scale them using traditional biotechnology? And that's where the synthetic biology comes in, where we can make small tweaks rather than the hundreds of changes needed to recreate these complex behaviors.
That is so smart. It's so very clever. That's awesome. So quick shout out to where you did your PhD and lab.
Yeah, so yeah, I did my PhD at MIT. So I was there when a lot of the seminal people in synthetic biology, so Drew Endy, the founders of Ginkgo, such as Reshma Shetty, et cetera, they were all there. There was a strong community, and we were kind of discussing how to engineer microbes, whether we should. And there was a lot of energy and enthusiasm, which I kind of guess swept me up. And so I remember when a lot of the next generation DNA assembly techniques, such as the Gibson method, came online, which I think is really powerful for making these large libraries of constructs. It was the early days of CRISPR, and we didn't really know what CRISPR was, but there was a lot of cool activity happening. And now, I guess apart from trying to understand these new systems, which is what I did in my postdoc. So after my time at MIT, I went to University of California at Santa Barbara, where I learned about systems biology, so learned about genomics and transcriptomics and how to sequence organisms. And now I'm trying to marry the two. So how do we identify novel systems, and then how do we apply the technologies of synthetic biology to them? The thing that I guess people find surprising is that is also non-trivial. So a lot of things that we've developed, they work well in E. coli, they work well in Saccharomyces, but part of the reason why there's exotic microorganisms that we don't study normally is because a lot of these tools don't work well in them. And so there's a lot of R&D that has to go into that as well.
Yes, I think tool building for biology is something that had been sort of under celebrated, but also having been at MIT in that era when we were all getting swept up in the possibility of building really powerful tools to engineer biology. There is a lot, I'd say, more focus on it, and great to have talented people like you, doing that marriage of those efforts. Did you always think you would be an engineer like this?
So this kind of engineer, certainly not. I mean, when I was a high school student and when I was entering college, I certainly loved science and math, and I knew I wanted to solve problems. But it wasn't until I had entered college that I even recognized that we could use biology to solve these kinds of problems. And so in one of my first experiences, I was working with Pichia pasteuris, so a type of yeast. People do a variety of things with it. And to be quite honest, it was so long ago, I barely remember what it was. And I was new to the field, so forgive me for my ignorance. But just the fact that the project, if I remember, had me introducing a new promoter that allowed control of expression of some protein or another that I think might have been like a bioproduct, like might have been like a protein drug or something like that. But just the ability that we could go in, manipulate the DNA and have the cell do something new, I thought was really, really compelling. And then again, and then as I learned more about it, the fact that not only could you use it to make things that are traditionally biologic, so like making proteins, such as insulin or other medicines, but we could begin to think about how do we engineer organisms that could grow into clothing, that could form new materials. There's a wealth of applications of biology beyond the conventional squishy things that we think about with biology, and I found that really, really fascinating. Mm-hmm. And so, which is kind of why we're trying to address these very hard problems of sustainability. So, yes. How do we get rid of plastics? That's not something I would have thought biology could address 20 years ago.
Right. I mean, I think, as you say, biology has had a lot of time to solve these challenges or challenges that are a lot like the challenges we face, and we could learn a lot from what biology has learned to do already. So that's pretty cool. So you always were a sciencey, mathy kind of a person. Did you have a family that was encouraging you to pursue this, or teachers who would encourage you to pursue this?
So my family was supportive, but they're not scientists, and so it really the possibilities I think came from really good teachers and mentors. And so I remember after I had kind of gone through this experience and realized that biology was really cool. One of my mentors, he convinced me to apply to graduate school. And I grew up in Canada. And so when he encouraged me to go to graduate school, I thought like, oh, I'll go to University of Toronto or I'll go to McGill. I mean, they're great places, but they're not MIT, they're not Stanford. But he's just like, no, you will work with the best people ever. Here's a list of places that you must apply for. After I was done laughing, I just decided to take the leap and do it. And I don't regret that decision. I mean, at the time, it seemed very overwhelming and very challenging, but I grew into that position. I mean, I think as humans, we thrive best when we're pushed, when we're stretched, and to do that, you have to learn how to be comfortable being uncomfortable.
Yes, it is so important to dream big, but it is also so discomforting, right? Like yes. You just feel like the sands are shifting under you. So how wonderful you had a mentor who encouraged you to dream really big and to push you in that direction. That's so important. I know that you spend a lot of time and care a great deal about mentorship and turning around and helping the next in line. You've done that with teaching that we did together, through MIT, and you've done that since you've left. Do you want to say a little bit about how you open your lab up to local students?
Yes. So just to kind of go back to that example with that mentor who kind of opened his lab to me and kind of showed me what biology could do. I've been very passionate about and a very strong advocate for student research. And so I am very willing to accept any and all students that show the right level of motivation. And so early in my career, so I mean, in the last, I want to say in the last 7 years or so, I probably mentored about 80 students, both directly in my lab or as part of the iGEM program. And I would say that early in my career, one of my most rewarding examples is a student who was definitely not an A+ student. He was a solid B minus student. But he seemed to be curious about this, and so I let him into my lab. And through that experience, he found that, hey, I really like biochemistry. I really like doing this genetic engineering. And he picked those electives, and certainly those classes were all A's and A pluses, and he like raised his GPA. He's now going to graduate school. He's now a graduate student. And I think just the ability to kind of spark that fire has been really rewarding. And so I mean, right now this summer, I have 5 undergraduate students, I have 2 high school students, and I mean, I really like to pay it forward and kind of share that same spark that someone was kind enough to share with me when I was their age.
Well, it is so incredibly important, and obviously something I deeply am committed to as well. So thank you for doing what you do. It makes such a difference. It just changes the future. It really does, and in such a positive way that will amplify and have all kinds of ripples. So I am so excited to see what you and your students will innovate and do from here on out.
Thank you.
Yeah, thank you for talking with us.